Okay, so on to chapter 4, which is our final chapter that we're going to cover in unit 1. So here we're going to talk about prokaryotes, and I'm going to give you a pretty brief overview. So let's just refresh our memories on how prokaryotes are different from eukaryotes. When we come down to it, it's really the way that their DNA is packaged. So prokaryotes, even though they're small and simple, they definitely have DNA. That's how their genetic information is carried, just like eukaryotes. The only difference is that eukaryotes have their DNA contained in a special compartment called a nucleus, whereas prokaryotes don't. They're really, really simple. Okay, the other difference is that prokaryotes that we're dealing with in this class, which are bacteria, have the chemical peptidoglycan, which we talked about in chapter 3, allows us to gram stain bacteria. But remember, peptidoglycan is only found in the bacterial cell wall. Okay, it's not even found in the other type of prokaryotes, which are the Archaea, which we talked about as the third domain. Prokaryotes, because they're so simple, also, they don't have organelles like mitochondria or, the smooth endoplasmic reticulum or Golgi bodies. It's basically everything just kind of floats around in the cytoplasm. So here are some things that we're going to talk about in this chapter. We're going to talk about different appendages or things that kind of hang off of the prokaryotes. And in this case, I'm really going to use the term bacteria because bacteria are prokaryotes, and those are the only prokaryotes that we'll be dealing with in this class. And then we're going to touch on these other structures as well. So when you talk about bacteria, they're really, really simple. And in fact, the most simple bacteria only have 4 parts or 4 structures. Okay, so bacterial cells, like every cell, has a cell membrane. Okay, now I want to make this very clear. The cell membrane is a different structure from the cell wall. Okay, so the cell wall is located outside of the cell membrane. Okay, so all of your cells have cell membranes, all plant cells have cell membranes, all bacteria have cell membranes. Then they have cytoplasm, which is basically, the kind of jelly-like substance that everything floats around in. They have some structures called ribosomes, which for the scope of this exam, I just want you to know that they're little protein factories, okay? So they're where proteins are made. And then they have their DNA. And bacterial cells, they have a chromosome. They don't look like our chromosomes. It's like a circular chromosome, and it's not contained in a nucleus. It kind of is tethered to the side of the cell membrane, kind of like if you think of a boat tied to a dock. So the boat can kind of, drift around a little bit, but it stays tethered to the dock. That's what it's kind of like, okay? So kind of floating around in the cytoplasm there. So most bacterial cells also have a cell wall, which contains the peptidoglycan and which is located next but more outward compared to the cell membrane. And also, most of them have that sticky coating on the outside. So for the scope of this class, I'm using all of these terms synonymously, okay? So if you see, like in your homework for chapter 4 or the exam or a quiz, envelope, capsule, slime layer, glycocalyx, lipopolysaccharide layer. For this class, I'm going to say that they all mean the same thing. They're basically that layer of gunk that's outside the cell wall, okay? And that can make the bacteria a little more pathogenic or a little more deadly. If you take higher level microbiology, you'll learn all of the different, kind of subtle biochemical differences in between all these different structures. So then we can go up another level, and these are structures that are found in some species of bacteria. So we're going to talk about all of these individually. Okay, we've already mentioned endospores, okay, that hibernation structure. We've also already talked about capsules, slime layers. Those can be structures that are found just on some bacterial cells. So this drawing looks big and scary and overwhelming. I just want to point it out because it's a good study tool. Don't panic that I'm going to have this on an exam and then make you label and describe all the parts. That's not going to happen. I just put it in there because it pretty much describes and labels everything. Okay. However, there is one structure on here that I want you to notice. So if you were to look within the bacterial cell, you kind of see this blue thing that looks like one of the Great Lakes. That is actually the bacterial chromosome or the bacterial DNA, and notice it's just kind of floating around. And then you have this smaller circular structure, and that is called a plasmid. Okay, so the definition of a plasmid is a small circular piece of DNA that carries, and this is really important, non-essential genes. So it's basically extra genes. So a bacteria cannot have any plasmids and just be absolutely fine. It's not going to hurt it. But sometimes they get those little extra pieces of DNA and they pass them back and forth, and that is one mechanism of how bacteria develops antibiotic resistance. Okay, so I just want you to be aware of what a plasmid is. So first, let's talk about external structures. Okay, appendages. Just like you guys have arms and legs, these are things that bacteria have that are connected to their cell wall and then just kind of hang off the outside. So some of them are there for providing what we call motility, which just means movement. And so I'll be talking about flagella because those are basically used for movement. And then the other part are used to allow the bacteria to stick to the host cells. Okay, so your cells, if you happen to get a bacterial infection, they can stick to each other. And then also a structure that is used for what I term here as mating, but realize bacteria don't mate in a traditional sense. It's not as if you have male bacteria and female bacteria. No one's producing sperm and egg. The closest they're going to come to mating is passing those little teeny tiny plasmids back and forth. It's the only way that bacteria are capable of exchanging DNA. Okay, so that's what I'm talking about. So we're going to talk about pili and fimbriae, which are involved in those processes. Okay, one term that I want you to understand is the term chemotaxis. Okay, and I'm a big fan of breaking down words and trying to get a simple explanation for them. So chemotaxis, if you take the first part, chemo just means a chemical, and then taxis means movement. Okay, and I remember that because, well, in the days before Ubers, you would take a taxi if you wanted to get somewhere. Okay, so that's how I remember it. So there are two different types of chemotaxis. Positive chemotaxis is when bacteria move towards something positive, like, oh boy, there's a whole bunch of, there's a really good food source over here, I'm gonna head this way. Negative chemotaxis is when bacteria move away from something that's bad, like, oh, there's an antibiotic there, I don't want that to kill me, so I'm gonna move away from it. Or, wow, there's too many bacteria here, there's no nutrition, there's a whole bunch of waste products, I'm out of here. That would be an example of negative chemotaxis. And make no mistake, we think of bacteria kind of sitting in little piles on our agar plates. You'll see that when you come into lab next time. But make no mistake, when they are in your body, bacteria are constantly on the move. That's how infections spread. So, there's going to be more and more of them. They're going to keep consuming energy in the form of, nutritional molecules that you're producing, and they're basically, eating you. And they're going to move away from things that are negative and move towards, more positive parts of your cells to be able to keep themselves alive. So that's what chemotaxis means. Okay, so next going to talk about a structure called pili. And the long fancy word is conjugation pili. So pili are these very rigid, long, basically tubes, and they are used in the process of conjugation. Okay, if you've ever heard a conjugal visit, okay, where a prisoner, and someone who's visiting them can get together and have sex. Some prisons allow that. That's what conjugation is. It's basically sex. Bacteria don't have sex. The closest that they get to sexual reproduction is when these conjugation pili form, and it allows bacteria to pass plasmids back and forth. There's no way they're going to pass their chromosomes back and forth because the chromosomes are way too big to fit inside these pili. And so, the best they can do is pass plasmids back and forth. Now, pili form completely randomly, so they could potentially form between two bacteria that don't have any plasmids, and then nothing happens. But let's say a bacteria out of, just spontaneous mutation has developed a plasmid that now contains a gene that can inactivate penicillin. Well, that's going to allow that bacteria to be, more able to survive. And if it starts passing that plasmid back and forth to a lot of different bacteria, you're soon going to end up with a bacterial population that is completely resistant to penicillin. Okay, so that is the main way that antibiotic resistant strains develop, is by passing these plasmids. And the plasmids end up, basically just kind of randomly by, random genetic mutation, end up with these genes that could potentially, allow a bacteria to be resistant to a particular antibiotic. Then there's fimbriae. Okay, fimbriae don't have anything to do with conjugation. What they are basically bacterial Velcro. Okay, they're little small bristle-like fibers, and they allow bacteria to stick to each other. Okay, that's why when you look at little bacterial colonies on your petri dish in lab, you're going to see they're actually little piles of bacteria, and it's the fimbriae on the outside that allow them to stick together and be able to form that little pile. It also allows bacteria to be the first way that they stick to your cells. When you potentially get bacteria, let's say in a cut or a wound, they use fimbriae to stick to you before they can start to reproduce and establish an actual infection. Okay, so this shows, this is taken by the electron microscope, okay, and kind of the dark gray structures you see here, those are the conjugation pili. And remember, there's no rhyme or reason to them. They just kind of, happen spontaneously and randomly. And then if you look at this little cloud, this dark halo, that is the fimbriae, okay? I'm gonna show you some better pictures where you can see what it looks like. This shows it a little better. So the things that are sticking off all around that are kind of short and bristly, those are the fimbriae. And then this long tube here is the conjugation pilus. Okay, this is even better because it's in color. So they're very small. The fimbriae are very small and bristle-like. They're pretty easy to tell apart from, say, flagella, because flagella are much longer. This is how E. coli in your intestine, which remember is good for you, is how it's able to cling to your intestinal microvilli. It's because E. coli has Velcro or fimbriae all over the surface. Okay, so now let's talk again about that layer of blech that's kind of on the outside of a lot of bacteria. And we're using terms like glycocalyx, capsule, slime layer, lipopolysaccharide layer. And it's on the outside of the cell wall, okay? So it basically can do a couple of things, okay? It protects the bacteria because it's gonna be a lot harder for things like antibiotics to force itself through all of that gunk and be able to get inside the cell. It also kind of has the same function as fimbriae, where it makes cells sticky, and so they can stick to each other and they can stick to you. Okay, so I don't know what this is. This is a stupid slide. I need to get rid of it. It doesn't really show anything. Okay, so functions of the capsule slash glycocalyx slash slime layer. Okay, so remember, the thicker that layer, the more pathogenic, or the ability to cause disease, the more virulent the bacteria tends to be. It also, because that layer tends to look kind of lipidy, which is what your cell membranes are made up of, it actually allows the bacteria to kind of fool your immune system into thinking, hey, this is one of our own cells, because it looks kind of lipidy, it kind of looks like our cells. It's not going to fool your immune system forever, but it can maybe buy that bacteria an extra 24 or 48 hours before your immune system figures out, hey, this is a bacteria, we need to get this out of here. So yeah, it will protect the bacteria against a type of immune cell that you have called phagocytes. And those are the types of cells that are in charge of identifying bacteria and viruses and basically engulfing them and eating them and getting them out of your system. So yeah, buys the bacteria a little bit of time. Also, as I mentioned, it's kind of like a moat around a castle. So it serves as a barrier to antibiotics and medications that need to get into the cell to kill them. And then it makes the bacteria sticky too. So it kind of does the same thing as Fimbriae. It allows the bacteria to stick to you and then also to stick to each other. Okay, the cell envelope basically does the exact same thing. Okay, just want to reiterate that, remember, the cell wall and that layer of peptidoglycan are what differentiates gram-positive bacteria from gram-negative bacteria. So remember, the gram-positive bacteria have a really thick layer of peptidoglycan. Gram-negative bacteria have a very thin layer of peptidoglycan, but then have this little extra outer membrane, which looks like your cells. And so gram-negative bacteria are actually much better at evading your immune system. Once again, not forever, but maybe it buys them an extra 24 to 48 hours. And that's a lot of bacteria that can be produced within 24 to 48 hours. So it gives them a head start on causing, a more serious infection. So we've talked a lot about the bacterial cell wall. One thing the cell wall does is whatever shape the cell wall tends to grow in, that's the shape of the bacterium. So if it happens to grow in a circle, you have cocci. If it happens to grow in a rod shape, you have rods. Happens to grow in a spiral shape, you have spiral shaped bacteria. So yeah, it determines the shape. The cell wall, because of that peptidoglycan structure, is actually pretty strong. And so it provides structural support for bacteria. And it has to do with that peptidoglycan content. Okay, so let me talk a little bit about peptidoglycan. So if we break apart this word, peptido means protein, glycan means sugar. So what it is basically layers of sugars or carbohydrates, and then they are held together by protein cross bridges. So one of the examples that I use is, if you've ever gone to, let's say, Menards or Home Depot and looked at fencing material, maybe you're familiar with a material called chicken wire. Okay, so chicken wire is used to, build kind of a flimsy fence, maybe to keep, squirrels out of your garden or rabbits or whatever. But the glycan would be like having several sheets of chicken wire laying on top of each other. And then the peptido part is pieces of wire that could be threaded through the holes in the chicken wire to hold all of the chicken wire layers together. That's basically the structure of peptidoglycan. Okay, don't memorize NAM and NAG. I'm not going to ask you about that. But so these would be the layers of the chicken wire. That's the glycan. And then you have these protein cross bridges that hold them all together. So when we think about the world's most perfect drug, imagine if you could come up with a drug that destroyed peptidoglycan but didn't affect our cells, right? Because we don't make peptidoglycan. Lo and behold, been there, done that, okay? So penicillin is literally the most perfect drug in the world because it is designed to attack bacteria, but not our cells. Okay, unfortunately, we way overused it, and so now we have plenty of penicillin-resistant bacteria. Plus, because there's no such thing as a drug that doesn't have a side effect, lots of people, myself included, are allergic to penicillin. So great drug, but can't take it because, you know, we could go into anaphylaxis. So what penicillin does is it doesn't prevent the glycan from being made. It doesn't prevent the protein from being made. What it does is it prevents the protein from coming in and stabilizing these sheets of sugar. And so instead of being a nice, strong, intact peptidoglycan molecule, all of this stuff kind of floats apart and the bacterial cell wall becomes very unstable. And once that happens, well, you can kind of pop the bacterial cell membrane like a balloon and it's easily ruptured and the cell dies. You actually have kind of a little type of penicillin called lysozyme. Lysozyme is most plentiful in your tear ducts. And so it, digests peptidoglycan. And so it prevents gram-positive bacteria from causing eye infections. Okay, so don't be afraid to cry. Release that lysozyme. It's going to help keep your eyes infection-free. Okay, so last lecture, when I talked about specifically acid-fast staining, I had talked about cell walls that weren't typical. So a typical bacterial cell wall can be gram-stained. But then you have these ones that are really thick and waxy. That is Mycobacterium. And remember, that is the bacteria that causes tuberculosis and leprosy, and we have to use an acid-fast stain to force that dye inside because of those thick, waxy cell walls. So Mycobacterium, you can't really call it gram-positive or gram-negative because it wouldn't even show up on a gram stain. You have to call it acid-fast staining. The other type of bacteria that I want to mention, and I know it's similar, they both have myco in the front, but mycoplasmas. Okay, so mycoplasmas are the one type of bacteria that are so incredibly small that you cannot see them with the microscope that you use in lab. I know one thing I mentioned in lab was that your light microscope, you're able to see 99.99% of bacteria. Okay, so you're going to see everything that can be gram stained. You're going to see Mycobacterium with an acid-fast stain. That's still going to be visible. Mycoplasmas are so small that they actually can't be seen unless you use an electron microscope, which most hospital labs do not have. You have to go to the University of Iowa or Iowa State to be able to find an electron microscope because it requires special expertise to run and they are super, super duper expensive. Okay, so mycoplasmas, they are really hard to diagnose. You basically have to do every type of staining. You do a gram stain if nothing shows up. Then you do an acid-fast stain if nothing shows up. Then you start thinking, oh, maybe this, type of pneumonia could be caused by a mycoplasma, and maybe we need to send that, patient biopsy out to a special lab that has an electron microscope. Okay, so mycoplasmas, the only thing I want you to get out of this, they are super small. Okay, so very, very small. They're basically, close to viruses in how small they are. So one of the main species, Mycoplasma pneumoniae, which causes a type of pneumonia that, you basically have to exclude everything else And then if you still can't find the source, it could either be mycoplasma or maybe it's a virus that's causing that pneumonia. But that's how small it is. Okay, so one more big thing that I want to cover in this chapter. First of all, here are our shapes, right? 3 main shapes: round, rod-shaped, spiral. Okay, one thing I want to mention is pleomorphism. Okay, for some reason, there are a few species of bacteria, and thankfully not very many, but as they age, they change shape. Okay, so maybe from 0 to 24 hours, they are cocci, if you look at them under the microscope. But as they get older, they elongate and they look like rods. Okay, or you may have a bacterial species that it's a rod from 0 to 24 hours if you were to do a slide and do a gram stain. And then for some reason, after 24 to 48 hours, it contracts and now it looks like cocci. Okay, so it's really important to remember that the best time to do a gram stain is between 18 to 24 hours after that patient's sample was inoculated onto an agar plate. Okay, and it's because you get the most uniform gram staining and you don't have to worry about this. Okay, you want to be able to know what shape that bacteria is when it is a young cell, because that is going to allow for proper diagnosis. Okay, so luckily not many species do this, but it's why gram stains, acid-fast stains, whatever we're trying to do, are the best time to do that is between 18 to 24 hours after the patient sample was inoculated onto the agar plate. Anything over about 24 hours, a bacterial cell is considered elderly and it's not going to give you the most accurate staining. Yay, okay, so we've reached the end of chapter 4. I'm going to get this uploaded, and this completes the end of the material for unit 1. Okay, thank you.